We put so much trust in our medicine. We expect it to heal us, no questions asked. But what if the very thing designed to make you better was also carrying, well, an unseen threat? Today, we're going to dive into the invisible battle that keeps our medicine safe, a fight against microscopic enemies you would never, ever expect. So, let's start with a kind of weird thought. What if your medicine was also a meal? A meal for tiny, dangerous bacteria that could be hiding inside. It sounds completely backward, doesn't it? But believe it or not, this is a very real problem. Some of the most common ingredients in our medicines, things we assume are totally fine, can actually become a food source for microbes. And that can turn a remedy into a real risk.
Okay, so what are these tiny freeloaders actually eating? Well, the menu is going to look surprisingly familiar.
Just take a look at this list. When we see stuff like preservatives, sweeteners, or flavorings, we think, oh, okay, that's just to make it last longer or taste a little better. But for a lot of bacteria and fungi, this is an all-you-can-eat buffet.
And this isn't just some theory, okay? This has serious, real-world consequences. Get this, some types of bacteria, like Pseudomonas, are so incredibly tough, they've literally been found munching on the preservatives inside eye drops. And that's caused some really nasty infections, right where people are most vulnerable.
And it really doesn't stop there. I mean, even something as innocent sounding as peppermint water, which is just used for flavoring, can create the perfect little home for bacteria and yeast to grow. So the first line of defense becomes pretty obvious. We got to find out if there are any of these unwanted living guests in the medicine in the first place.
So if these little microbes can be hiding pretty much anywhere, how in the world do scientists actually go looking for them? Well, that's the first step of the whole investigation. The main tool for the job is something called the microbial limit test. Think of it as a crucial checkpoint for things that don't need to be perfectly sterile, like, say, syrups, creams, or pills. The test basically asks two simple questions. 1, is anything alive in here? And 2, if there is it one of the really bad guys we know about?
So that's step 1. It's an absolutely essential check, answering that one vital question. Is there anything alive in here that really shouldn't be? But what happens if the coast looks clear? Because the intruders are already gone. And this is where the story gets really, really interesting. See, the problem isn't just about what's alive. It's also about what the dead leave behind.
Exactly. Just imagine this for a second. The bacteria were there, but they got killed off during the manufacturing process. You'd think, great, problem solved. But when some bacteria die, their cell walls break apart, and they leave behind this toxic footprint. This footprint has a scientific name, a pyrogen. And it's a great name, actually. Pyro means fire or heat, and gen means to produce or generate. So it's literally a fever producer. And when these little things get into your bloodstream, your body can spike a fever, even though there are no living bacteria anywhere to be found.
So how on earth do you test for the ghost of a bacterium? Well, the answer comes from two very different and pretty remarkable corners of the animal kingdom. For decades, the go-to method was the rabbit test, which is exactly what it sounds like. You'd inject a sample into a rabbit, and then you'd wait and see if it got a fever. It worked, but it was slow. Then came this revolutionary alternative, the LAL test. Here, instead of a rabbit, you just mix the sample with an extract from horseshoe crab blood. And if there are pyrogens in there, the liquid instantly turns into a solid gel. It's a clear chemical signal that's way faster and much more sensitive.
And this, to me, is the most incredible part of this whole story. The LAL test, which stands for Limulus amoebocyte lysate, is 100% dependent on the amazing copper-based blue blood of the Atlantic horseshoe crab. You see, its blood cells have this special clotting agent that is just exquisitely sensitive to those toxins left behind by bacteria. It's the crab's natural defense mechanism, and for us, it's become a revolutionary safety tool.
When you just step back and think about it, it's pretty wild. The safety of the entire global pharmaceutical industry, all those injectable medicines, relies on a rabbit's potential fever
and a prehistoric crab's incredible blue blood. It's this hidden partnership between our own ingenuity and some of nature's most surprising secrets. And it really makes you wonder, doesn't it? What other hidden worlds, what other incredible bits of science and nature are quietly working behind the scenes to keep us safe every single day? Thanks for joining us on The Explainer.